Hi, welcome back. What I want to do in this video is I want to explore the structure of ATP synthase. We're going to kick off our understanding of ATP synthase with looking at its structure. And what I've done for you is I've written the very general reaction. So we're going to take ADP and ligate it to phosphate. So the reaction is basically going to be this. I'm going to have my adenine up here, right? Bend it there for space. And I'm going to have ADP, right? I'm going to have ADP. Have negative charges at physiological pH. And I'm going to put in a phosphate, right? I'm going to put in a phosphate, right? And essentially what's going to happen is the enzyme is going to facilitate the nucleophilic attack. And let me do the electrons in a different color. It's going to facilitate the nucleophilic attack of one of the, and actually this is the, just for nomenclature's sake, this is the alpha phosphate. This is the beta phosphate, and if it was ATP, the third phosphate would be the gamma phosphate. So this right here, this is going to become the gamma phosphate. And so what the enzyme is going to do is it's going to facilitate the nucleophilic attack right here. The electrons kick up, they kick back down, and water is expelled. So water is expelled, right? And so you end up forming ATP, and that's the general reaction of ATP synthase, right? So why don't we look at this membrane that I've drawn, and what we'll do is we will first of all we will label the space. Up here is going to be the inner membrane space, and down here I'm going to label as the matrix. So in other words, the outer membrane is up here somewhere. That's the outer membrane. Okay. Now what we have to understand first is that ATP synthase is functionally divided into basically two different parts, and those parts are the F sub 0 and the F sub 1 subunits. The F sub 0 subunits are part, or at least are situated in the inner membrane. So essentially what you're going to have, and I'll do this in a different color, you'll have this right here, okay? And then you're going to have, and I'll go ahead and label these in just a minute. And let me go ahead and draw these. And again, this is testing my artistic skills, right? Well, what did I just do? I think I changed the color. But anyways, so the F sub 0 subunit is the part that's really in the membrane of the mitochondria, and that's the inner membrane. So what do I have? Well, this stuff right here is the C subunit, and there are multiple C subunits. There's actually a lot of them in there, and as we'll see, this subunit, or this group of subunits, actually plays a very critical role in the actual rotation of ATP synthase, right? This red one over here is the A subunit, right? That's the A subunit. And these green ones over here, these long arms, are the B subunits. And there's actually two of them. And actually, I might also mention that this, in terms of the C, there's 10 of them, right? But in terms of the green one, the B subunit, there are 2 of them. So there's one on either side, and there would be one situated behind this one, right? Okay, and so the F sub 0, and let me do this in gray, the F sub 0 would be essentially all this right? It's the stuff that's located in the membrane. So basically the B subunits span both the F0 and the F sub 1. And what you'll find is that things that are not situated in the membrane, that delve into the matrix, are ultimately part of the F1 part, or part of the F1 subunit. or the F1 group of subunits, right? Okay. Now what I have, I'm going to do this in a different color, I have a shaft that sticks out here. A shaft that sticks out, and this is the gamma shaft. And it sort of sticks, it goes up all the way into the C subunit, and then it's going to sort of come down here, and I'm going to sort of draw dotted lines because it's going to go into something we call the alpha 3 beta 3 spheroid, right? So what is the alpha 3 beta 3 spheroid? Well, it's essentially so let me do that. Alpha 3, beta 3. So what you're going to have is, let me draw the alpha subunit here. There's one alpha subunit. And here is a beta subunit, right? So here's the beta. The beta I will do in blue. The alpha I will do in purple. And then I have another alpha subunit. And then I have another beta subunit. And then another alpha subunit, and then finally another beta subunit. And so basically you can sort of think of these 6 subunits. They're 3 alpha, 3 beta. They're sort of in a circle around the gamma shaft. And ultimately they're stabilized to the, well, they are part of the stator. And actually let's define the stator and the rotor. A rotor means that it's part of the moving part of ATP synthase. Well, the moving parts of ATP synthase are the A subunit, excuse me, not the A subunit, the C subunit. The C subunits actually rotate along with the gamma subunits. Okay? The C subunit and gamma subunit are actually what's rotating. And part of the molecule that functionally are important to the stator, which is the part that's not moving, is the A subunit and the alpha and beta spheroid at the bottom. Okay? And as we'll see, what ends up happening is the gamma shaft rotates against the alpha 3 beta 3 spheroid, and ultimately this is what produces ATP. And so let's kind of get a feel as to what's happening right here, right? So all these cytochromes, right, what did they do? Well, they pumped protons into the intermembrane space, right? Well, ultimately, they create a concentration gradient. They create a pH gradient, a charge gradient. And so what's going to happen is the protons are going to move back through ATP synthase, and they're going to specifically move through half channels in ATP synthase, and we'll look at that in more detail in the next video, but they're going to move through ATP synthase and power the gamma shaft rotation. So the gamma shaft is part of the rotor. The alpha and beta subunits are part of the stator. The C subunit is part of the rotor. And you could consider the A subunit as part of the stator, because technically the A subunit, although it's required for the rotor function, is not actually moving. The A subunit is just sitting there, and of course the B subunit would be part of the stator. And there's also an epsilon subunit, and I'll go ahead and draw that in here. There's somewhere in here there's an epsilon subunit. And at this point, I don't think they really truly understand what it's for, although it's been hypothesized that it's to inhibit the ATPase activity of ATP synthase, because actually ATP synthase is a reversible reaction. Notice when I drew this reaction up here, where did I put it? I put it up here. So I put ADP goes to phosphate and ATP, and actually, as we saw with the organic mechanism, it's with an expulsion of water, right? But you can actually reverse this reaction, and ATP synthase can do it. It can actually hydrolyze ATP with water to ADP and phosphate. So the epsilon subunit is thought to play a role in protecting the ATP from hydrolysis, especially in times of when you're needing to synthesize ATP, not hydrolyze it. So this is the general structure of ATP synthase, and this is the general reaction. Of course, we looked at the organic mechanism here. It's basically just a nucleophilic acyl substitution mechanism. So I hope this video gave you a little intuitive sense on what ATP synthase is. In the next video, we're going to actually look at the mechanism of the rotational catalysis. And then we're also going, in the next video, we're going to look at actually how ATP is synthesized directly. See you in the next video.